Okay, so I've already told you how DNA in the cell looks before mitosis happens. It's in long strands called chromatin, but during mitosis this changes quite a lot. So in the first stage of mitosis, it happens in 4 stages. The first stage is called prophase. And in prophase, the chromatin basically condenses and forms these chromosomes, which I showed in another video. It forms the chromosomes, and these are very short, they're condensed and they're thicker, and you can actually see them under a light microscope when they're in the prophase stage. The nuclear envelope, which contains the chromosomes, also breaks down in prophase to allow spindle fibres to come in and pull the chromosomes. So this also breaks down. The next stage of mitosis is called metaphase. And in metaphase, the spindle fibres come in from opposite poles of the cell and attach to the chromosomes. They then pull the chromosomes to line up along the equator of the cell. And they will line up in a random order, but this is vertical. So they line up vertically, and this can be in any random order. And the spindle fibres are what do this. This is very obvious in an exam. If they show you a picture of metaphase, you'll be able to see it because it's all lined up along the equator. That's metaphase. The third stage of mitosis is anaphase. This is also very obvious in an exam if you have this image, because anaphase is where the spindle fibres contract and pull the identical chromatids apart from each other to opposite poles of the cell. This is so that exactly the same genetic information is in each of the new cells. The spindle fibres pull the identical chromatids apart to opposite poles of the cell, and that's anaphase. It's very obvious because you can see that they're clearly been pulled. In telophase, which is the final phase of mitosis, it can also be called cytokinesis, which is where the cell is actually now pulling apart. So the chromatids have come apart to opposite poles, and now the cell is also pulling apart. So the cytoplasm will split into the 2 new cells. And eventually the nuclear envelope, which you can see at the top of prophase, the nuclear envelope will also reform in telophase. So in telophase, you can see that exactly the same genetic information is in each of the new daughter cells, because the sister chromatids are identical. They have identical DNA in them. It's simply that they have been separated. In another stage of the cell cycle, these will be replicated in a stage called synthesis. So the 2 new daughter cells will be exactly the same.